Friends, welcome to Biology Made Easy. Today in this video, we'll discuss the transport in the cell membrane. In the previous video, we have already started regarding this transport in the cell membrane, where we have discussed that the transport is mainly of three types: passive transport, active transport, and bulk transport. So out of these three, already we have discussed the passive transport in the previous video. So today in this video, we'll discuss about the active and bulk transport. So the transport across the cell membrane, it is mainly the passive transport, which already we have discussed, and the second one is active transport, and the third one is bulk transport. So already we have discussed it in the previous video. Now we will discuss about this active transport of substances across the cell membrane. The cell is an open system. Always substances from outside they move inside, from inside they also moves outside. It is a continuous process. And this cell never attains equilibrium. This also, you know, if it will attain equilibrium, then it cannot, it will have no life. So it never maintains equilibrium. If we'll give an example, it will still more clear. In a cell, if this is the inside of the cell and this is the outside, then you'll find that the potassium ions, they are present up to 100 millimolar inside the cell, while outside they are present in 5 millimolar. That means there are more in concentration potassium ions inside the cell, while there are less 5 millimolar outside the cell. Now if we take the case of sodium, then they are only 10 to 20 millimolar inside the cell. This is inside, this is outside. While they are up to 150 millimolar outside the cell. That means inside the cell there is more amount of potassium, 100 millimolar, while outside there are only 5 millimolar. In the other case, the sodium, it is more in the 150 millimolar outside the cell, while within the cell it is only 10 to 20 millimolar. Now how will you account for this equilibrium? Because your equilibrium is not maintained. So how will account for it? What is the answer? If you tell this is taking place in diffusion, then it is not possible. Because in diffusion, always diffusion will continue till equilibrium is maintained. But here in both the cases, equilibrium is not maintained. So how this concentration in one side is greater than the other side? It is due to active transport of substances. So that is why in active transport, what happens? If we will discuss the different factors in active transport. In active transport, the most important thing is from low concentration, it can move to high concentration. That means uphill movement. Uphill movement. That means the ions can move from low concentration to high concentration. That is uphill movement. That as we have seen here. Now this is the first point. The second point is, this is possible only by expenditure of energy. So for this energy is required, and for most of the cases, usually energy is ATP. And the third thing, for transport of substances, certain carriers are required, and these carriers are mostly the proteins. So the third thing is protein carriers. Protein carriers. So these are the three things required, which are just opposite to that of the passive transport. In case of passive transport, from high to low, but here low to high. In case of passive, energy was not required, but here energy required. And in case of passive transport, the the transport doesn't require any carriers, but here carriers are required. This is the difference between the two. Now, this active transport is also a vital process. It is a vital process. So, how can we prove that it is actual vital process? It has been seen that if there will be deficiency of oxygen, then the absorption of substance will decrease. That means decrease in oxygen decreases absorption. This is one point. The second point is all of you know that cyanide is a poison, and if cyanide is applied to a cell, it dies. So the same thing happens here. If cyanide is given, there is no absorption of substances. Cyanide inhibits absorption. Similarly, just like the enzyme substrate reaction, if substrate similar to this substance will be there, that will also inhibit. So the third point is structures similar to substances inhibit it. Then the next point is this temperature. If there will be increase in temperature, then the rate of absorption will increase. So effect of temperature, and it is a more rapid process than diffusion, and it takes place against the concentration gradient. So from this, it is clearly seen that it is a vital process. That means living cells are involved for all these cases. Then now the next thing is whether the carriers are involved or not. How can we prove it? Again, we can prove it from different evidences for the existence of carriers. Carriers involved in the process. So the first thing is we can take that is saturation kinetics. That means if you go on increasing the amount of solute, then there will be increase in the amount of substrates. That is the more and more will increase, but only up to a certain extent. So what is that extent? Till all the carriers present in the cell membrane are saturated. After that, if you still go on increasing the substrates, there will be no further increase because all the carriers are saturated. So that is one proof that the carriers are involved. Now we will take the second case. That is again that is specificity. All the substances are not absorbed equally. It is found that certain substances are absorbed in more amount than others. Because the reason is there are separate carriers for separate substances. So those in which there are more carriers, they are absorbed to more extent than others. That is known as specificity. And the third one is inhibition. Since the carriers are proteins, if we give certain inhibitors, since proteins are inhibited, so then naturally the absorption of substances are inhibited. So from this it is clear that in active process carriers are involved and it takes place only in the living cells because it is a vital process. Now let us give different examples. Now the active transport The active transport is mainly of two types. It may be primary active transport, and the other one is secondary active transport. What is the basic difference between these two? In case of primary active transport, the metabolic energy ATP is directly utilized for the transport of substances. But here the metabolic energy or ATP is not directly used. It is accompanied with some coupled reactions. When energy is used for some other reaction, so it will take place at that time. There is no direct metabolic energy for this. That means for this type of transport, there is always a coupled reaction. This is the basic difference between the two. Now let us discuss the primary active transport. Then we will discuss the second one, that is the secondary active transport. So for the primary active transport, there are several examples, but the most important is sodium potassium pump. That is the most important one. So the primary active transport, when I will give different examples, the first example is sodium potassium pump. And for this, for work in this sodium potassium pump, the James Kuo, Japanese scientist, was awarded Nobel Prize in 1997 in chemistry, because he worked on this sodium potassium pump in the cell membrane of crab, and for this he was awarded Nobel Prize. So what is the sodium potassium pump? The sodium potassium pump, it operates in most of the mammals, and it usually operates in case of the nerve cells and also in muscle cells. Nerve cells and muscle cells. These are the common examples. Now what happens usually? Now if suppose this is the cell membrane. This is cell membrane. This is the cytosol side, and this is outside. This is the outside. Now here the ATP, energy ATP, that is this is the ATP. It will be broken by the help of enzyme that is ATPase. Now when this will break, what will happen? There will be exchange of potassium and sodium ions. That means for expenditure of one molecule of ATP, usually 2 potassium ions come inside and 3 sodium ions go outside. 3 sodium ions go outside. That is for expenditure of one molecule of ATP, 2 potassium ions come inside the cytosol. They come inside while 3 sodium ions go outside. So as a result, what happens? That means the concentration of the sodium becomes more outside, as I have given you in the past, this example you see. The sodium concentration is more on the outside, while the potassium concentration is more inside. As a result, what happens? And this takes place about that is 300 sodium ions per second and per pump. At this rate, the pumping takes place. So when the sodium ions go outside, that means outside the cell, always the outside cell always it is positive. It is always positive, while on the inner side of the cell it is always negative. It is always negative. That means always one that is resting, one potential is maintained. One potential is maintained. And out of the total amount of ATP that is formed in a cell, about one third of ATP is spent only for this sodium potassium pump. That means this is so much important, and these things take place in case of nerve cells and also in case of muscle cells. That is why the more amount of sodium are outside, it maintains a positive charge outside and negative charge inside, so that always it maintains a potential across the cell membrane. So this is the sodium potassium pump, which is very important for this. Now, what is the importance of this sodium potassium pump? That will be discussed. What is the importance? Now, number one, you will write here. Let me write it here. Importance. Now, the first importance, it maintains positive charge outside the cell. This is a very important point. Then this is important for nerve impulse conduction. The third point, that is muscle contraction. The fourth importance, that is you can tell that is urine formation in kidney tubules. Then another important point is, in case of the aquatic animals, or if you take that of the marine birds like seagull and penguin, they drink that marine water which is salt. That is why they have a nasal glands through which they remove the excess amount of salt. That means the next example is, you can tell that is marine birds, and the example you can take seagull and penguins. They have nasal glands, and through the nasal glands they remove sodium. But how the chlorine is that moves once the sodium goes actively, then chlorine is passed passively. So sodium goes actively while chlorine moves passively. So these are the different importance of this sodium potassium pump. Then there is a second example. The second example is that of the calcium pump. The second one is calcium pump, and this calcium pump usually it operates in two cases. The first example you can take that of the RBC. In RBC the calcium pump operates. That means from the RBC calcium moves outside. That is calcium is calcium to outside. That is from the RBC. Similarly, we will take the second example. The second example is in case of muscle cells. So in muscle cells, usually what happens, there is a sarcoplasmic reticulum which help in muscle contraction and relaxation. So what happens? Suppose this is a sarcoplasmic reticulum. This is a sarcoplasmic reticulum. So this is sarcoplasmic reticulum, and this is this side is a cytosol. Now, when from this is cytosol, that is a calcium will move into the sarcoplasmic reticulum from the cytosol. So this will lead to muscle relaxation. And when this calcium will come out, here calcium enters, so it leads to relaxation. When from the sarcoplasmic reticulum calcium comes out, then it leads to contraction. That means whether the sarcoplasmic reticulum is present in muscles, That means whether the muscle cells will contract or relax, that depends upon the whether if calcium is moves into the sarcoplasmic reticulum muscles, relaxation. If comes out, then it is contraction. So here this is known as calcium pump, and it requires also, that is energy, active process. Now let us discuss one third example also.